Okay, why don't we get started now? Just to remind you, so I'm going to teach you about the copy number variance here on the somatic side, and then we'll do a hands-on exercise to get you to play with it for a bit. And then Emma will come over and talk about the germline copy number, which is very different, very, very different than the somatic one. This workflow, I'll tell you, has so we have it's described in a paper and on the GATK work on our website. There's a lot of math involved in this. Today's presentation, I'm going to keep it very high level. I'm going to describe generally what's happening with the math. And if you're interested in more details, feel free to go to the website or the paper or anything you want, or even the code. You can go look inside of it if you want. But we'll try to keep it very high level so I don't scare people. I'll also warn you that the somatic workflow is very complicated. All of it is put into a WDL to make it easy. And again, you're welcome to see how we do that and download it and run it in your own bash script or whatever you want. Okay. Okay, so the copy number variance, again, this is generally, these are just large insertions or really large, I should say, not insertions, large duplications or large deletions. Duplications and deletions. Structural variation includes these, but there's a lot more. But for today, we're just going to talk about copy number variance. And tomorrow, Emma will teach you about structural variation, the more complicated things. We've been working on copy number variance, not just in the GATK side, but in many, many teams, many tools for a long time. And it's been hard. It's actually taken a while for us to do well. Early on in the days of SNPs and indels, we got success quickly. It took a lot more with the copy number. And that's because, again, you have very short reads, and there's so many inconsistencies, and there's so much noise. We are just making a lot of mistakes. I'll actually mention the first time in the 1,000 Genomes project. So this is a project from around 2010 and 11. We used to do validation. The first time we ever validated any inversions, every single one were wrong. So we must have called 100, 200. Every single one we validated was wrong. And so in the early days of copy number and structural variation, we really didn't do a good job. So it's taken a while. What we've shown here, this plot is kind of over time we've improved things. And so now at this point, I would say between both somatic and the germline side, the tools are much, much higher quality. I think Emma will talk to you about some actual clinical cases on the germline side. The germline colors used in our clinical pipeline for rare diseases on the germline side, and it's used very effectively. If you ask the researchers, they are very happy with their performance there. Whereas if you asked them 3 years ago, they would have said they cannot make copy number calls. So we've really done a great job of improving things. And the same is true on the somatic side. The somatic world, we've had a longer history, and the tools have progressively gotten better too. So if you've seen any of the names of any of these tools in the past, you shouldn't be surprised. But the thing we're going to show you today, I'll talk about model segments, which is true both for exomes and genomes. But for the most part, the plots I'll show you are exomes, mostly, because most of the cancer stuff is exomes. And then Emma will talk to you about GCNV after I'm done. Again, also for exomes and genomes. Showed you this picture before, so I won't go into too much detail here. But remember, this is why it's hard to call copy number variance, because the copy number is difficult. I'll just give you the case again in chromosome 4. You might here you have not just 2 copies, but you have 3 or 4 copies. But the third copy is only half a copy, only half of the chromosome. And the fourth copy is also only half and also has other stuff attached to it too. This is what's being sequenced. And this is why it's hard to figure out exactly how many copies we have. I talked a little bit about TP53 yesterday, or BRCA. I just want to explain to you why it's so important. I will reiterate today why this is so important. So there's 2 types of genes that are implicated in cancer. There are tumor suppressor genes and oncogenes. A tumor suppressor gene is a gene that does a good thing, and gets turned off by cancer. So TP53 was an example. Again, that is in charge of killing cells that are misbehaving. And when it gets turned off, the cells don't die. BRCA was another good example. That gets, we mentioned, that is used to repair damaged DNA. And when that gets harmed, mutated, and we have problems there, or overregulated. It could be nothing is happening to TP53, but when MDM2 is overexpressed, you have 7 copies of MDM2 instead of 2, then you overregulate TP53. Okay, so that's when a tumor suppressor gene is turned off. An oncogene is a normal gene that, when it gets duplicated, turns into something bad. Okay, so things that encourage replication, for example, so that's the top there. There are lots, genes that might encourage cell reproduction that get turned on. So it encourages that too often. That's an example of a oncogene. An oncogene is something that in its abnormal process does something bad for the cell. Does too much. It's overexpressed. And so both of those things are very important to understand the progression of cancers. And when you look at medicine for clinical trials for patients who have cancer, a lot of those clinical trials, a lot of the drugs that they produce are aimed at specific mutations, specific genes. So just as an example, there could be a drug that targets TP53. So will help the cell, if it'll notice that TP53 is knocked out, and will help the cells regenerate the protein that's produced by TP53 as an example. So knowing the mutation that's driving your particular cancer helps you figure out which drug to take and which clinical trial to go into, for example. That makes sense? Every cancer is different. Unlike germline, which is inherited from your parents, every cancer shows up and it's all different. Someone actually mentioned in the break, it's a good point here. There's something called a cosmic database. Cosmic is an example. I think, Aaron, I don't know if you mentioned this at all. Yeah. So Cosmic is a database of known hotspots where there's mutations. But it doesn't mean, unlike Nomad, where you've seen this in many different participants, in cancer, you don't expect the same mutations in 2 different patients necessarily. Maybe the same genes are normally knocked out because they're so important, but you don't see the exact same mutations. And that's much different. Okay, so knowing what's driving your cancer, God forbid, someone else's cancer, patient's cancer, is very important in the treatment. And that's why the sequencing is important. So back at home, we encourage anyone who is diagnosed with cancer immediately to get sequenced, to get their tumor sequenced, because that helps drive clinical care. There's an example here on the bottom of what it looks like on the germline side, but I'll let Emma talk about the germline rationale for copy number 2. There's a lot of good stuff there. And so I'd mentioned this before. We're going to go into details now. Just as our toy example. So again, in a normal, you always expect this is 2x coverage, but in general, represented here, imagine you have 2 copies, one from your mother, one from your father. In the cancer, as we saw in that cell, you don't have it. And you also have this imbalance, right? So if certain copies can get duplicated, and then some of them can get mutations, and some of them don't get them. So you no longer expect a 50-50 ratio on the SNPs, and we'll talk about why that's useful for copy number at the end. But also the ratio in terms of your coverage is going to be far off. So the whole point of this exercise is to find differences in coverage. Now, when Emma talks about structural variation, we'll do more than just looking at coverage. But for the CNVs, for duplications and deletions, we're basically just looking at coverage. It's not exactly true. I'll show you that for the somatic stuff later. But for the most part, we're looking at coverage. Good? All right, so here's an example of an exome on top, and there's different kits here, and a genome on the bottom. The genome is pretty easy. Let's start with that. It's not exactly the same coverage all across the genome. For the most part, a nice PCR-free genome has pretty consistent coverage. And it's usually consistent from sample to sample in terms of your average. So if you're at 20x in chromosome 1, you're usually around 20x in chromosome 2. And the same thing is true across participants, different patients. It's not true with different capture kits with exons. And I mentioned this. Different capture kits have different effectiveness at different exons, different places in the genome, depending on GC richness, depending on how many mutations are in that exome from that sample, can cause differences. So everything is very different. And you can see as an example here, very different profiles. Look on the middle one, the 2 exons, one has a lot more coverage than the other. Now, this doesn't necessarily mean there's a copy number event here. This is what we have to contend with. This is what we deal with. And this is where the math is going to target. Clear? Good. All right. So we're going to talk about there's copy number and copy ratio. Copy number is an absolute thing. Usually you say there's a extra chromosome. So instead of copy number 2, it's copy number 3. All right. That assumes integer-based things. Remember that picture I showed you before? Things aren't necessarily integers. And so we can't always assume that that even though one part is copied in this copy, maybe only half of that was copied in another. So there's actually fractional copies we can have. And so the easiest thing to do is to talk about things like copy ratio. And so the ratio is going to be relative to normal. So again, if your copy number is 2 and that's your diploid, your copy ratio, that means is 1. It's the percentage you are relative to 2. That makes sense? So the pictures will show you actually 1 is normal going forward. Okay, we're going to start talking about copy ratio, where 1 is this baseline from now on. So 1 means you're diploid, and anything more or less is a difference. And this is how we deal with non-integer values in the somatic side. In the germline side, you don't have to deal with that. Germline, it should always be integer, unless Emma tells me I'm wrong later, but it should be. Okay, and then we also talked about purity here. So this is the bottom right. I talked about this earlier, but again, it's very important. Imagine you have 3 cells. 1 is from the normal, and then 2 are different from the tumors. Right? This is what's very common when you pull out cells from a tumor. You get a mixture. It's very heterozygous. And so notice that A is a SNP that's expressed in 2 out of 10 total copies, and B is only 1. So you see different ratios. Okay, and so here we call up the tumor purity is 2 out of 3, which means that every 2 out of 3 cells are actually the tumor, but that means 1/3 are actually in the normal. So you can have a lot of contamination here. And that's not sample-to-sample contamination. It's just your cells contaminated. So you're trying to deal with all those things. But just remember that when I talk about copy number, we're talking about copy ratio for the pictures. The 1 will be our normal state going forward. Okay, but when we eventually make calls at the end, we'll go back to thinking of things as copy number 2 and 3 and 0. All right. So now I showed you this plot before. I've turned it back on the right axis. So the x-axis is the position in the genome. Okay, and every dot here is 1 exon or 1 capture bait. Okay, inside your exome. And we're plotting based on your copy ratio. So this is relative to average. So you should be on average at copy ratio 1. But look at, it's all over the place. This is normal. So look at the normal sample. There's no tumor here. But relative to baseline, to your average 1, you have many, many, many exons that have a lot higher or lower coverage than your average is. And that's the noise, because this is normal. They should all be 2, again, or 1. Sorry, they should all be copy ratio 1 here. And then in the tumor, you see a very similar thing. So this is all we get. This is the data we get. We just start with. We get these reads and the counts, and from this, we have to figure out where the copy number is, where the differences are. All right, who can tell me where the copy number events are here? I will buy you a really nice bottle of scotch if you can tell me from this picture without cheating. Okay, it's impossible. It's impossible. So now we're going to show you the mathematical equations used to remove all the noise. That's what we're going to do for the next few minutes. Okay. Again, I'll try to keep the math high level, but it's important to know what's going on and play with it. You don't have to do the math. The GATK does the math for you. But we want to teach you what we're doing so you understand it. Okay, so there's 3 steps, and I'll go into each one in detail. But this is just a high level view. Collect data just means get the read counts. for every exon, every position, get those read counts, make that plot, denoise it. That means kind of normalize it, get rid of a lot of the differences, and then find the actual segments. Find the pieces that are different. 3 steps. And we're going to do that each. I'm going to show you how the command lines work. We'll go into details for all of these. Okay, remember I told you the workflow is a little bit more complicated? So the half-dotted caller was one tool. CNV calling is not one tool. And we have 6 steps. We're going to go through them all. Some of the steps, by the way, I should tell you, are preparatory. You only have to do them once, like creating a panel of normals. You only do that once, hopefully. Okay, but I'll still teach you how to do it. But there are 6 steps if you're starting from scratch. But once you have your panel of normals, you don't have to do it again. We'll go through all of them at once. Okay, so the first thing you need to do, okay, is process intervals. Everything is parallelizable here. So again, we're going to imagine your, I'll focus mostly on the whole exome case, but we have a whole genome in there too. Okay, so when you have a whole exome, you want to treat every exon differently, really every bait, every target differently. Okay, and keep that as a separate data point. So we also there's a lot of information in the reads beyond the actual targets though. So what we do is we create an interval list with padded on 250. So we have 250 bases on either end of the baits of the targets, and use that information. So first we're setting things up so we can parallelize it all the way, and also not look at reads that are not near the exon. So by the way, I should explain to you, this is important. When you do exome sequencing, it's also not perfect. So you will definitely get what are called off-target reads. Reads that aren't, even though they're not part of the baits intended, they still show up. We don't want to count those. Those are clearly not expected. So this helps do that. In the whole genome, if you don't create intervals, you're basically going to treat every chromosome as one contiguous interval. So those plots I made, you won't have lots of points. You'll have 23 points, one for each chromosome, basically. Okay, so what we do is we break it up into groups of about 1,000. And that can change, but we do that here. Every 1,000 bases along the exome is another point. And that gives us a resolution. If you want to have more resolution and find things that within 100, that means you can find smaller events. But you get a lot more noise that way. If you know you're expecting larger events, you can make this 10,000. So to be clear, again, this is your resolution. How much I'm breaking up the genome in, and so this means every 1,000 bases is considered 1 point. Okay, so I'm not going to be able to find events smaller than 1,000 here, but that's okay because most of my events are massive. This is the command line. You've seen the GATK command line many, many times. Again, but just to say, the first line shows you the name of the tool. The R is the reference file. An L, the -L, is an intervals. I don't know why I called it L. I don't remember. It was a long time ago. But L tells you the intervals to use. You want to send it for an exome, the exome targets. And for the whole genome, you don't have to send it anything. Okay. XL on the genome tells you which intervals to exclude. So it says, don't look at the centromere. There's a blacklist. A blacklist means these are places telomeres, centromeres. We're not going to find good copy number in there. Those are just repetitive elements. Don't find them. So that's why it's there. And then we talked about the padding also there. Okay? And your output is going to be an interval list. So this gives you the intervals in terms of our plotting, tells you your resolution here. Clear on what step 1 is? Okay. You only have to do this once. Just once. All right. Now we get to collect our read counts. So for every one of those intervals we just identified, let's count the average coverage in those intervals. And that's how we create those plots. Good? So here on this toy example here, given that this is a given one interval, let's count the average depth in there, and we put that in there. So on the bottom right here, you see approximately, if you followed the same command lines we ran in the previous slide, you'd end up with about 150,000 targets in an exome. All exomes are different, but that's about what you find. And in the genome, you break it up into 11.5 million bins. Okay, so that's a lot of data points. It's pretty good. Clear what's happening so far? Because it's going to get harder. All right, good. This is the command line for it. The command line should be pretty simple. If you're wondering what this interval merging rule is, overlapping only, just means this is whether if there's two intervals that overlap, you would merge them. But really, you could ignore it for now. So again, this takes in a tumor. By the way, we're not going to have a matched normal necessarily for this, but this will take in a tumor, for example. It'll take in the intervals from the previous step, from step 1, and output this HDF5 file. This is the header. It has all the counts for every one of those 11.5 million or several 100,000 points from the previous one. It has one data point for each one of those. Simple, right? We're just counting up reads now. Easy. Now we can make that, at least that first plot. All right, panel of normals. We've talked about this before. So you've definitely heard of panel of normals. If you don't know, you lose that bottle of scotch. Okay, but I will say again, just like the panel of normals is important for SNPs and indels, it's even more important, I think, for the copy number. The panel of normals is used to remove artifacts, noise. And I showed you how much noise there was. There's so much noise here. So we have to create a panel of normals, again, one time for copy number 2. And what this does is it tells us across many samples, this particular target, this particular interval, I see it's generally at higher coverage than it normally is across everyone. So when I see in my tumor here at high coverage, that's actually not different. That's normal. Okay? Or I see that this target is always at low coverage in many different samples. So when I see it low coverage in my tumor, it's not necessarily a deletion. So it's very important to have a panel of normals in this. And that's how you remove noise. Okay, so given the panel of normals, you use that, you run principal component analysis, just a mathematical tool, and that's how you denoise. You can think of it as I like to call it normalizing, but it's denoising. It's removing all that noise. Okay, by taking what we see in averages across different samples. We'll talk a little bit about the panel of normals coming up, but this is important. You get that? The most powerful tool you have in copy number, somatic copy number calling is that panel of normals. Without it, you cannot do copy number calling. Good. So how do you create the panel of normals? And I'll say once more, you only need to do this once for your sequencing type. Once you change to a new sequencing machine, you go from the HiSeqs to the NovaSeqs or NovaSeqs to the NovaXs, you should create a whole new panel of normals. When you have a different capture kit, a whole new panel of normals for that one. Because again, you want to be as consistent and as similar as possible to your tumor sample. Right? So you take in, the way to do this, so just like we made read counts for we do it here. We create read counts for many, many, many samples. So -I is your input. You can input 1 file, 2 files, and a whole ... as many as you want. Just take in the counts, and it'll run an average and store it and create your panel of normals for there. Okay. There's another argument you can use for GC correction if you want, but that's a little bit advanced. Just to remind you, GC bias means that part of the genome has a lot of G's and C's and doesn't capture or sequence well. So we can model that too. So this is in creating your panel of normals. Pretty simple command line. Just your inputs and one output. The hard part of creating your panel of normals is actually finding the data, finding the things to use. But once you have that, it's a lot easier. Okay, and then we talked about the denoising. So once you have a panel of normals, going from the really ugly blue plot to the nice green clean plot, removing the noise, normalizing, this is the command line to use. It's actually not such a hard command line. So again, it's called denoise recounts. It's a tool in GATK. You have your input. This is the counts from the tumor. Okay. And these are outputs sorry, these are going to be outputs, okay? And again, if you want to use your denoising with GC correction, you can do this. If you want to play with expert command line options, you can. We're not going to do it in my tutorial here, but there are lots of tutorials for you to look up online. I won't go into the expert stuff, but it's a pretty standard and easy command line. So the issue isn't the complexity of the command line, it's just how many steps there are. Okay, so again, just to remind you, this is taking the panel of normals and taking the counts and making them normalized, denoised. Good. I'm trying to go slow so people can ask questions if they want. So this is what happens. This is what that step does. Okay, so again, you have the really noisy, messy stuff that are raw. You've done some standardization, and then you've the principal component analysis. Now, when you look at the bottom plot, notice that for the most part, the genome is at copy ratio 1. That's expected. Copy ratio is, again, your ratio relative to diploid, to 2. And then there are a couple of large events you see that stand out. The way to read these plots, by the way, is this is chromosome 1, and then it changes color to 2, 3, 4, 5, 6. So right in chromosome 7, most of chromosome 7 here looks to be at copy ratio, probably 2. Okay, so copy ratio 2 means how many copies of chromosome 7 are there? A little bit of math. Aaron has the right answer in the back. You're cheating. So come on, someone yell it out. Copy ratio 2 means there are how many copies of chromosome 7? No one. Thank you. Just 4. Thank you for doing it. Okay, so there are 4 copies because it's not ratio 2 means you're 2 times 1, and 1 is at your standard is at 2. So 2 times 2 is 4. So this means there are 4 copies of chromosome 7 over here. And this looks like you're at copy ratio 1/2, so you're at 1 copy. 1/2 of 2 is 1. That's the way to think about this. At some point, we'll make real calls, but this is the way to look at this. Nice, right? Okay, we're almost done. This is the hard part, and the steps are pretty easy. There's just a lot of things. Sometimes it gets more complicated, and the better the panel of normals, the better you can do. Now look at whatever chromosome this is. A lot going on here. Now, what this is saying, actually, and this is hard for you to just try to visualize this, is that, again, you had 2 original chromosomes, one from your father and one from your mother. One of those has been duplicated, and one of those was deleted. So for the most part, your copy ratio is still 1, but you have 2 completely different chromosomes. Okay, you have 2 from your father and none from your mother. And that's why it's messy like this, and that's why we still have more steps to do. But this is what something like that would look like. Pretty cool, right? Scary, although that actually happens in tumors, but this is what you see. Okay, so this is just a plot showing you that the better your panel of normals is, the larger it is, the more accurate it is, the more close it is to the type of sequencing or capture, the exact process, the better. So even if you have the exact same sequencing machines here in Taiwan that we have at the Broad in Cambridge, if we make the panel of normals for you, it'll be close, it'll be good, but it won't be as good as if you made your own panel of normals because our labs are different and the people running them are different and they're different. The sequencing machines are different, and who knows, there's more dust. We have more dust and we're on the ocean, near the ocean, like who knows? Right? Okay. So the larger and more closely you are to the tumor data, the better you'll do. Okay. This was just because the panel of normals is so important. People say, oh, how many do you need? Do you need is 1 enough? Is 2 enough? Can you use the normal of the sample? So we try to, I don't need to read everything off the screen to you all. You can all read it. You have these slides, but there are a few things I wanna highlight here. One is people had asked, I think Aaron earlier, what about chromosome X and chromosome Y? That's really important with a copy number. Because again, chromosome X are very different, males and females. Chromosome Y, very different. So if you're we don't often call chromosome X and Y, it's complicated. We don't know in our sequencing pipeline what the gender of the samples are. And if we don't want to make assumptions either, because if we can't just look and say, oh, it's copy number 2, because they're all somatic. So I don't know, we don't know if they're real or not. So we cannot make those assumptions. We don't want to make false calls. So we usually don't call it in chromosome X and Y. But people still should. You should want to. We just can't. So if you do that, you'll need a separate panel of normals for males and females, because the baseline is different for males and females. And it'll also help you find cases of XXY, for example. All right. What else? People had asked how many samples you need for a panel of normals. It is different for SNPs and indels versus copy number. For copy number, anything is good, but we found 40 to be a good number to be a minimum. Try to hit 40 if you can. Okay. And it all depends on how noisy your data ultimately is. So that's just a recommendation. Don't take that as 40 as the answer. But we found that works pretty well for us as the minimum threshold. And one really important point I want to hear is this middle bullet right here. When you take normals, people ask me, can I take the normal sample from my tumor? Usually, yes, you actually can. There is some contamination with a tumor in the normal, but for the most part, you can take normals of your tumor samples, unless the tumor is a blood-borne tumor, or if it's metastasized. Then you cannot. Okay, so you have to be very careful about that. But for the most part, we actually do build a lot of panel of normals from the normal tissue of the tumor patients. All right. Okay, so that's a review. We've gotten through most of that. In the top part, we've processed intervals. We've gotten read counts. We've created a panel of normals. We've denoised, normalized. Okay, and now we're almost done. We actually haven't made calls yet. We've just gotten those cool plots. We have to finish up. So let's finish up the process. Okay, that's that last piece there. There's also some things we can do with SNPs and indels. Let's teach you about that. The nice thing about copy number is we can use the information from the SNPs and indels you used with, for example, from UTECT to learn some stuff. So look at the example we have up top. This is just, again, a toy example. You have in our copy here, we have 2 copies. We actually have 2 copies of the reference, the wild type, and 1 alternate one. So we have an extra copy, basically. We have 1 extra copy of the chromosome. So when we say alt, what I mean here is that there's a whole bunch of SNPs on that one. Let's say there's one with lots of SNPs on it. Okay, and the one without all the SNPs has an extra copy. So 2 things have happened. Lots of somatic mutations have occurred on one of them, or there even could be germline ones. There could be lots of germline mutations. Okay, so one copy has, let's say, 1,000 germline mutations. And the other copy is all reference. This is just for a toy example to understand. And the one with only the reference one with no mutations gets duplicated. Just explain to you what's going on. So what you'd expect to find in the original copy, it should be a allele fraction of 50%. These SNPs should all show up 50-50. But in this case now, because the copy without the SNPs has been duplicated, they should all show up at an allele frequency of 1 and 3 now, 33%. Okay? We can use that information. If we see a whole bunch of SNPs in a row at 1/3 frequency, that gives us a good hint that there's some sort of copy number event here. Now, in the germline side, that's bad. Something is going on. Or it could be a germline event too, a germline copy number. Okay? But in the somatic case, these, again, the copy number events are so large that we can see over a long swath of DNA, a long area, the same 1/3 ratio for heterozygous SNPs. For homozygous SNPs, that obviously this doesn't help. Does that make sense? So we're going to teach you how we do that. You're going to take in your tumor. That's the case fam. Case and control. So control is normal, and case means tumor. Okay, and control, and count your counts. Okay, so you're basically going to count your case and count your control. because we're looking at the differences here of ratios. And we're going to run it between there. So we're going to do 2 things now. This allows us to both do the denoising and also the modeling of SNPs that look like they have a balance, allele balance that's different. Allele balance means that their ratio is not 1 and 2 to 1 and 3, or 2 and 3, or 3 and 4, or 1 and 4. And again, if we see a lot of that over the same region of the genome, it gives us a good hint. So we use both pieces of evidence to be able to call events. Is that clear? Important. Yeah, I'm getting some nods. Okay. All right. Now here's the hardest part. So sometimes the important thing here is that in those pictures, you didn't have a lot of resolution at the ends. I couldn't tell you exactly. So we showed you some easy ones where all of chromosome 7 was duplicated, or all of chromosome, but clearly it's not always true that all of the chromosome is. You want to get the breakpoint. You want to know where that ends. And that's what the segmentation is, figuring out where the pieces are. And it gets complicated. I just want to go back in your head to the original picture of chromosome 4, where that 4th copy had only had a fractional duplication. So we want to know where sometimes you go from copy ratio 4 to 3 to back to 4, back to 3. It's all over the place. So we need to figure out exactly where those shifts are happening. And that's what the segmentation is, figuring out where the groups that are together and different are. Make sense? And so that's what we're trying to do here. And I don't need to go into the math or anything. So this is a kind of slide showing that the old way of doing it and the older tools was not great. It was not accurate. But now we figure out how to take both. Again, those allelic ratios, the 1/3 down to 1/4, back up to 1/3, down up to 1/2, really helps you figure out where those segments are. Okay? And so that's what this is saying. So this is the command line. You'll get to do this yourself soon, very soon. I'm almost done. You'll get to do this yourself. But again, you're now gonna create. So this allele counts is something we created in the previous step. So that's going to be an input also into modeling, and you'll both your tumor and normal allele counts. Okay, and then there's some like advanced command line arguments we're showing you here that I'm not going to go into, but you can play around with it at your own point. Okay, so this is the command line, and we'll let you run it very soon. Not super complicated. These are just the inputs from previous steps, and this is the output we're going to output. Here we go. This is an example of what you might see. One thing I'll say is that sometimes you have more noise in your data than you'd expect, and I'll show you some example. You'll want to play around with things potentially a little bit, but this is an example from a whole exome where some do better than others, and they change around a penalty factor to try to get resolution. So sometimes the parallel way of thinking about this is you're trying to have a projector, and the projector doesn't have the right resolution. So you kind of change the knob a little bit to bring the resolution in and out. The same thing is true here. So in the projector, because how far you are, you don't have great resolution. We do the same thing. We try to adjust the knob a little bit with this penalty factor to try to get better resolution. And you're seeing that in some of these slides. So the different things you're being seen have different resolutions. And in some of them, you can see some pretty cool things happening. Okay, so this is from the copy ratio. And this is after we've segmented things, you can see that this is actually very blurry, right? But as we get more and more resolution, we start to find out exactly where the differences are. Each one of these is a different segment. And so you'll see here, it's very messy. Your resolution is very bad. We turn the knob a little bit, and you're getting a little bit better. And you turn the knob better, and you see here, this is copy ratio 1, 2. No, we're at 1. Okay, here, copy ratio 1/2. Okay, and then all of a sudden, what happens? You've got something happening. You get one of the chromosome has now both duplicated and been deleted. Fun things. Okay, but you only see that as you tune the resolution. So sometimes with copy number calling, you do have to rerun things a little bit, and that's why we give you the tools to plot things so you can see it yourselves. I don't know if they'll be plotting in the hands-on, Probably they'll be doing some plotting, right? We'll find out. Anton says a little bit of plotting in the hands-on coming up. All right, so I think this is the end of the workflow. You see what we've done. We've gotten those allelic counts to help our segmentation better. We've modeled them, and then we've made those segment calls. That's the overview for the CNVs. And the last thing you have to do is, given those segments, we're actually going to make calls. Pretty simple, okay? The tool that takes in those segments from before and outputs the calls. I don't think there is a easier, a more basic GATK command line than this one tool. There's an input and an output. We take those segments we found and actually make calls. I have to say, I don't remember if this outputs a VCF. I think it does not output a VCF. The cancer world has been slower to adopt the VCF format. Aaron will tell me that's a good thing that I was not involved. But the cancer version of VCF is even worse. So, don't mind me. Ultimately, though, with copy number, you don't expect that many. It's not like you expect billions and millions of copy number events in a tumor anyways. So none of these files get particularly massive. So notice here there's 11. So 11 has multiple segments. So in all of chromosome 11, you get a bunch of 3 segments. So we're not making that many calls in the genome. And this is going to be the final step, and you'll get to do it yourself in the hands-on and make your own calls. And again, what you should see then is here, there's one call for these chromosomes, one here and a little bit here, and not too many. All right, so we've gotten, and also we have the ability to make all those plots. So you'll want to see the exact same plots that we have been showing. Those weren't just special for the workshop. Anyone can make those plots. It's part of the GATK package. By the way, people complain that the GATK Docker image is so massive. It's because we got to include R and Python and all the things to make these plots. So it's not our fault. Blame R. And then, so this is, if you wanted to plot, you'd run these command lines. And again, I'm going to go quickly because you have these slides. They're all in a folder you can look at. They're online in documentation. I think they're even in the hands-on. They're in the tutorials. They're all over the place. So if you ever want to run this yourself, okay, so you use these to plot things. And that is it. So we've gone through the entire workflow. It's 6 steps. There's a lot. Again, unlike the haplotype call or even MUTAC2, where there's 1 or 2 steps, here there's a lot of steps. But I wanted to show you, make sure you understood the rationale for everything, so you know why you're doing things. In the upcoming hands-on, you'll have a chance to make copy number calls all on your own. I'm going to stop now and take questions. When we're happy, we'll have Anton come up, or if you even want to come up now, Anton, and prepare the hands-on, feel free to. Before lunch, Anton had you start up the environment for the next hands-on. So I'll stop. Any questions about any of that? Yep.
The panel vector, like how to increase.
Yeah. So are they going to adjust the penalty? Yeah, it's okay. Are they going to adjust the penalty factor in the hands-on? Do you know?
Nope.
No. Okay. So you're talking about this here, and so I think this is usually the default works. This is just kind of showing you when you have I'm like right in the. So let me stand right over here. Sorry. This is just showing you what happens when as you adjust the numbers. Usually you'll just start with a standard one. If you started with a standard one and you get to something, the default in the GATK, and you get something that looks like this, you'll plot it yourself and say, wait, this doesn't look good. Then you'll increase yourself a little bit each time. It's a very quick process. It doesn't take long to run. Again, there aren't these we've used, now we're on to tens of thousands of intervals. So it's a very quick process. Plot it, show this. If you see something that looks like it has bad resolution, increase it a little bit. That's the way to do it. Okay. And so I wouldn't recommend starting with 0.05 necessarily. We're just showing you what it looks like at that ratio. I don't remember what the default is, but you'd start with a default and increase it as you need it. Okay. Now, you wouldn't just start at 5 necessarily, because it means you might miss smaller events. So that's why you don't do that. So getting the balance is right. Usually the default option is the right one. Good? Okay. Other questions? I'll wait like 30 seconds for anyone from the other room to come by if they want. Feel free.
It on text.
Okay, feel free to get set up on there also. Okay. In that case, I will hand the microphone over to